Thank you, Chloe, and thanks to Jane as well for the opportunity to talk here today. And unfortunately, we do have a few tongue twisters in our deck, so I'll try and get through those without too much pain for everybody. So Cynata is a clinical stage biotechnology company based in Melbourne, and we are focused on the development of a revolutionary manufacturing platform which we call Cymerus. Now, the reason it's revolutionary is this platform allows us to make an effectively limitless number of therapeutic stem cell doses from a single blood donation. And that's particularly important because it is this is the major obstacle to commercialization of these highly promising therapies, and I'll explain a bit more about that in a moment. So we've already generated some really compelling clinical data using our cell therapies, and again I'll tell you more about that momentarily. And we have a rich clinical pipeline at this point with 4 ongoing clinical trial programs. And importantly, 3 out of those 4 trials are randomized controlled trials that are going to have results readouts between early next year and early 2026. So there's a huge number of very important inflection points coming up for the company. And we're also about to start a new clinical trial in patients with kidney transplant very imminently. So the clinical data I was mentioning just now. Our first clinical trial was conducted in a condition called graft-versus-host disease, or more specifically, steroid-resistant graft-versus-host disease. Many of you may not have heard of this condition, which is fortunate because it is a really terrible condition. It occurs in patients who have received a bone marrow transplant or similar procedures, and it arises because immune cells in that transplant recognize the recipient's body as foreign. So it's almost like somebody has somebody else's immune system inside of them. And you can imagine how horrible that is. The main treatment initially is steroids. And in patients who don't respond to steroids, it's called steroid-resistant GvHD. And unfortunately, the outlook for those patients is very, very grim. And sadly, most of them pass away. And that's the population that we ran our clinical trial in. So these patients had already failed to respond to steroids, and at the time that was the only treatment available, so things were not looking good for them. We treated 15 patients, and we saw really encouraging results. So 13 out of 15 showed an improvement of at least 1 grade, and 8 out of 15 showed a complete response, which means that the GvHD resolved entirely. And we saw a 60% 2-year survival rate. And while 60% might not immediately sound that great, in the context of this patient population, it really is. So in other papers published around that time, based on other therapies, the survival rate at 2 years was around about 20%. So clearly that's a very encouraging outcome. And importantly, we also saw no safety concerns. And another interesting thing about this trial is it was the first ever completed clinical trial worldwide using what we call iPSC-derived cells. And I'll explain what that is in a moment, but I think in recognition of that milestone, the trial was published in Nature Medicine, which is one of the leading medical journals worldwide, and it was also featured on the front cover of that month's issue, which really highlights what an important trial this was for the wider field. Now, sorry for showing these pictures in the middle of lunch, but the other trial that we've announced data in is in diabetic foot ulcers. These are chronic wounds that occur on the feet or lower limbs of patients with chronic diabetes, and they're very difficult to heal. It's a major problem. It often leads to amputation and really impaired quality of life in these patients. This trial has now completed recruitment, but the initial data we announced earlier this year were in the first 16 patients at the 10-week follow-up point. And what we saw was that the extent of healing in the stem cell treated group, and this is a stem cell based wound dressing, so it's a very novel product, and the extent of healing in that group was close to 90% after 10 weeks, compared to just over 50% with standard of care dressings. So again, really encouraging results so far from that trial. Now, just to explain what our manufacturing platform is and why it's important. So a conventional process, and I should clarify here, MSC means mesenchymal stem cell. This is a type of stem cell that's very useful and has been investigated widely over the past 15 to 20 years for lots of different medical conditions. A conventional process involves getting people to donate tissue, for example, bone marrow or adipose tissue or other sources. They then isolate the MSCs from those donations, but they then have to grow the cells in a lab, which is a process called culture expansion, in order to generate enough cells to do what they want to do. Now, that has a lot of major challenges. First of all, it's logistically challenging and costly to have this endless need for new donors. But more importantly, there's actually a huge amount of variability between these donors. So the MSCs produced from these different people are different. They have different levels of activity, so you end up with an inconsistent product. And that's a huge problem with a pharmaceutical-like product, because the regulators require you to show that your product is going to be consistent from batch to batch and year to year. Another issue is when MSCs undergo this extensive expansion process, they change, they lose potency eventually and become less useful. And our process overcomes these challenges. So by using this iPSC technology, we can make all of the cells we ever need from one donor, one time. So we have this cell bank, which has the capacity to reproduce itself effectively limitlessly, and we then have proprietary technology, which we licensed from the University of Wisconsin-Madison in the US, which is a world-leading center for stem cell research. And that technology allows us to turn these iPSCs into MSCs and manufacture them that way. So by doing this, we can completely avoid the need for new donors. So we avoid all of that logistic and cost challenge. We avoid the variability between donors. And we also avoid the need to excessively expand the MSCs, which, as I mentioned, causes those cells to lose functionality. So when you put all these things together, what this means is we have a way of making consistent, high-quality product in a scalable manner. And that's what's really required for commercialization. So just to give you a little overview of MSCs, the reason these cells are useful is they have a lot of properties, including they balance or modulate the immune system, and they also promote tissue repair and regeneration, and that's why they're useful for so many potential clinical conditions. Now, the advantages of the iPSC platform, I should explain finally what iPSCs are. It stands for Induced Pluripotent Stem Cell. And these are adult cells. In our case, they come from a blood donation, so they're blood cells that are then reprogrammed to behave like embryonic stem cells. And I'm sure everybody knows we all arose from embryonic stem cells. Embryonic stem cells themselves have been utilized in research, but there's a lot of ethical controversy associated with that. So what we do is we produce adult cells that behave like embryonic stem cells, and that means they can reproduce themselves to an effectively limitless extent, and they can turn into any other cell type. So they're an ideal building block for manufacturing cell therapies at large scale. I'll quickly mention that we have a partnership with Fujifilm, a strategic manufacturing partnership. Fujifilm is a major player in the manufacture of advanced therapies such as this. Most people probably just remember them from the manufacturer of photographic film and so on. Obviously, that's not a very lucrative business anymore, and they've diversified into all sorts of other things, including this industry. Our pipeline, in the interest of time, I won't go through the rest of this in too much detail, but again, please do download the full presentation and you can read more of this at your leisure. But we now have a phase 2 GvHD trial that's ongoing. This is a global trial taking place in Australia, the US, and Europe. We recruited the first patient in March this year, so we're actively recruiting patients now. We anticipate completing recruitment by the end of this calendar year, and if we do that, we should have results from this trial in the middle or early second half of next year. So that's obviously a very exciting one. Our diabetic foot ulcer trial is the one that I mentioned earlier. As I said, this has completed recruitment, so the final patient visit in this trial will be in September this year, and we'll get the final results shortly thereafter. It'll be, we're not quite sure yet if it'll be this side of Christmas or just after, but either way, not too far away. We also have a trial in osteoarthritis, which is in partnership with the University of Sydney and funded by the Aussie government. This is a phase 3 trial. It's a randomized placebo-controlled trial in 320 patients. Again, this is fully recruited. We're expecting the last patient visit in November next year and results shortly thereafter. This trial is seeking to show a disease-modifying benefit of these cells. In other words, the cells don't just help with the symptoms, but they help to actually stop the osteoarthritis process, which I'm sure everybody knows means you have this degeneration of joints in your body, and it's a huge problem. Right now, there are no disease-modifying therapies approved for osteoarthritis, so if we can show success in this trial, it will be absolutely huge. And finally, our kidney transplant trial is expected to commence imminently. Again, this is in partnership with an academic group and funded externally, this time in the Netherlands. The idea here is that by administering the MSCs, we can reduce the dose or even avoid using immunosuppressant drugs. And the reason that's important is these drugs help to stop the transplanted kidney being rejected, but they're really, really toxic and they cause huge problems. So if we can move away from those, it would really revolutionize the way transplants are handled. And with that, I will let you read the rest at your leisure. Actually, I'll just briefly mention we are pursuing a partnering business model, which means we're looking for partners who will either in-license our existing clinical assets, but also potentially take on our platform and start pursuing other applications of that platform, really to maximize its value. And we have a number of catalysts coming up, as I said. Later this quarter, we'll have the start of the renal transplant trial. Later this year, we should have some results from the initial cohort in that trial and complete the GvHD enrollment. The DFU results will be late this year or early next year. Then we'll have GvHD results in the second half of next year, followed by osteoarthritis results in early 2026. So there's a huge amount coming up. And with that, I think I'm slightly over time, so as I said, I will leave you to read the rest at your leisure. I will also be here for the rest of the afternoon, and I'd be delighted to talk to anybody who wants to find out more. I believe my contact details are available, so I look forward to hearing from you. Thank you.